Hello everyone, and thanks for coming. I'm Visnovska, a bioinformatician based at Aarhus University Hospital in Norway, and I will introduce you to pipelines for processing sensitive sequencing data which we develop in our department. The department I'm part of is called Department of Clinical Molecular Biology, or EPIGAN for short, and it's a joint operation of Aarhus University Hospital and the University of Oslo's Institute of Clinical Medicine. EPIGAN does its own research, but it also provides service and support, including bioinformatics, to external clinicians and scientists. In such a hospital-based research environment, many high-throughput sequencing experiments are performed on human samples and lead to generating sensitive data. In order to appropriately store and analyze such data, we use one of the Norwegian computational infrastructures dedicated to work with sensitive data. It is called TSD, which stands for Services for Sensitive Data, and includes access to high-performance computing cluster. One of the features of this cluster is that a user doesn't have access to internet from within. However, Singularity software is available, and that allows us to use containerized bioinformatics tools and data resources. As availability of the sequencing data increases over the time, so does the number of research groups around our department which use the data in their research. This results in a demand for reusable software to perform analysis of next-generation sequencing data, which can be easily deployed in independent project environments of the cluster. The presented set of the pipelines is our attempt to provide such a software using Snakemake for orchestration of the pipelines and Singularity for containerization so that the pipelines themselves do not depend on the software installed on the underlying computational infrastructure. Let me move a bit farther down the poster now. In this middle panel, you can see the list of currently available pipelines. At the moment, there is one pipeline for analyzing ATAC-seq data, another one for ChIP-seq data. I'll skip the feral flea painter for now. The fourth one is called MADE, and it serves for analyzing metatranscriptome data. And the last one is for analyzing RNA-seq data. Let's get back to the feral flea painter. It uses outcomes from ATAC-seq and RNA-seq pipelines to perform differential analysis of those data. The results of the two types of differential analysis are then merged by the pipeline to explore the relationship between the differential gene expression and differential accessibility. To access the EPIGAN GitLab page, you can use the QR code located in the right-hand bottom corner of the poster, or simply use the provided URL. Let's now move up the poster a little bit and zoom into one of the pipelines. We are looking at the MADE pipeline now. The pipeline rules are shown as bubbles, and the dependencies between them are represented by arrows. We introduce a rule hierarchy here, with the high-level rules being located at the bottom of the figure. All the rules related to one high-level rule share the same color and are executed together with the high-level rule if need be. An analysis is guided by a sample sheet file that indicates which files are associated with one sample, or with different sequencing lanes of one sample, or different read types. During the initial step of the analysis, which is shown here in the first column, we have a rule called check presence. This rule crunches through the sample sheet file and creates empty presence files, one for each sample, sample line combination, and so on. This makes it easy to use different subgroups of input files for different rules. For example, FastQScreen rule runs separately for each lane, while merge lanes requires all lanes for one sample as an input in order to merge them. Most of the time, we use BioContainers Singularity container images to make bioinformatics tools available in the pipeline. When such an image cannot be used, we provide definition file for the purpose-built container image as part of the repository. For example, functional biocontainer for Kraken is available, while Metaflon container needs some post-installing steps which we have to be handled manually, and thus we had to build it on our own. At the bottom of the poster, you can find all the bioinformatics tools used in these pipelines. At the end of the presentation, I would like to acknowledge the importance of TSD in the context of handling sensitive data. Thank you for watching and looking forward to any questions.